Bismillahirrahmanirrahim. So our new topic of discussion is glycolysis. The word glycolysis itself is a self-explanatory word, which means the lysis or the splitting of the glucose molecule. It's a metabolic process where a glucose molecule is split down to make the two molecule of the pyruvic acid, and these pyruvic acid molecules are then ultimately they make acetyl CoA, which then ultimately joins the Krebs cycle, and in this way they synthesizes the energy for the cell. This metabolic process actually happens in the cytosol part of the cell, while the Krebs cycle, which is the downstream part of the glycolysis, although it's a separate metabolic process, but it's a downstream process of the glycolysis. The Krebs cycle actually happens in the mitochondria. While the glycolysis, where the glucose molecules are stepped down, the glucose molecules are stepwise break down into pyruvic acid molecules. It happens in the cytosol or cytoplasm part of the cell. Generally speaking, in the glycolysis, actually the glucose molecules are stepwise undergoes through the different process of either the cleavage or the phosphorylation, and ultimately they form the pyruvate molecule, and then the pyruvate form the acetyl CoA, and then the acetyl CoA join the TCA cycle, tricarboxylic acid cycle, or the Krebs cycle. So in the very first step, the glucose is phosphorylated to make glucose 6 phosphate. So there are two choices for the glucose 6 phosphate, depends upon the condition. Whether the glucose 6 phosphate carry on to join the glycolysis, or it may also join the pentose phosphate pathway. So the glucose 6 phosphate pathway, if it decides to join the glycolysis, it is then converted to fructose 6 phosphate. The fructose 6 phosphate, this step is basically a reversible step. So here either the fructose 6 phosphate, either it goes down to make a fructose 1,6 biphosphate. It means that the fructose 6 phosphate is further phosphorylated. So this fructose 6 phosphate, either it may go down to make a glycerol 3 phosphate, or it may again make the fructose 6 phosphate. So when the fructose 6 phosphate is again phosphorylated to make a diphosphorylated fructose 1,6 bisphosphate molecule, so it splits into two 3 carbon compound, because fructose is 6 carbon compound. So it splits down to make a 2 molecule of 3 carbon compound. One is dihydroxyacetone phosphate, the other one is glyceroldehyde 3 phosphate. So the dihydroxyacetone phosphate, it ultimately makes the glyceroldehyde 3 phosphate. So dihydroxyacetone phosphate ultimately makes the glyceroldehyde 3 phosphate. The glyceroldehyde 3 phosphate is again phosphorylated to make a 1,3 diphosphoglycerate. It is then dephosphorylated again to make 3-phosphoglycerate. Then in the next step, the position of the phosphate is changed from carbon 3 to carbon 2 to make the 2-phosphoglycerate. And then this 2-phosphoglycerate make the phosphovinyl pyruvate. And the phosphovinyl pyruvate, then it makes the pyruvate. And from this point onward, it makes the acetyl CoA, and the acetyl CoA joins the TCA cycle. So the end product of the glycolysis is the pyruvate. So it's, roughly speaking, it's a 10-step process, starting from the glucose and it ends at the pyruvate. So now this slide is basically explaining a stepwise preparation, stepwise splitting of the glucose molecule, along with the structural overview that how the glucose molecule is stepwise converted to the different intermediates of the glycolysis. So generally speaking, the glycolysis is converted into is divided into two phases: preparatory phase and the payoff phase. In the preparatory phase, basically the phosphorylation of the different intermediate happens, and there basically they utilize the ATP to make the activated molecule, which in the payoff phase split down to make to produce the energy. So that's why the first 5 steps are called preparatory phase, because they prepare for because they invest the energy by taking up the ATP, and then in the payoff phase they produces the more energy. So in the preparatory phase starts from the glucose. The glucose is first phosphorylated by utilizing a single ATP molecule to make the glucose 6 phosphate. Here you can see that this one is the glucose molecule and this one is the 6 carbon, so here the phosphorylation occur. Then the glucose, and this first step is mediated by the hexokinase. You can see here the different enzymes. Every enzyme is labeled with a specific number, and here with each of the step a specific number within the circle is basically tagged with each reaction, which shows that this reaction is mediated by enzyme number 1. Here's enzyme number 1, hexokinase. This step is mediated by enzyme number 2. Here is the second enzyme, phosphohexoisomerase. So in the next step, glucose 6-phosphate is converted to fructose 6-phosphate with the help of phosphohexoisomerase, because glucose and fructose, they are the isomers of each other. So what happens in this, basically it changes the isomerase, it changes the functional group. Glucose has the aldehyde functional group while the fructose has the ketone functional groups. So here it happens that the glucose is converted to the fructose 6-phosphate. Then again in the next step, another phosphate group is added by utilizing the ATP to make fructose 1,6 bisphosphate. And this reaction is mediated by phosphofructokinase. Kinase is the enzyme which adds the phosphate group. Then this fructose 1,6 bisphosphate is split down or to cleave to make two 3-carbon compounds. Here you can see the color discrimination. This one is the fructose molecule. So when it's split down, it is split in this way, exactly splitting the fructose 1,6 bisphosphate into two phosphorylated 3-carbon compounds. One is the glycerol 3-phosphate, the other one is dihydroxyacetone phosphate. So the dihydroxyacetone phosphate, it ultimately changes to the glyceraldehyde 3-phosphate basically. And this reaction is mediated by the aldolase. Uh, the triphosphate isomerase. This fourth reaction is mediated by the aldolase, which converts the fructose 1,6 bisphosphate to two molecules of 3-carbon glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this one, so the last 5 steps are then the payoff phase, where the high energy compounds are formed. So here the energy producing steps, it takes place basically. So glyceraldehyde 3-phosphate, it is then again phosphorylated to make 1,3-bisphosphoglycerate, and in this step basically the NAD is reduced to make the NADH. The reduced form of the NADH is the energy rich compound. It upon oxidation produces the energy, the ATPs basically. Then the next step, 1,3-bisphosphoglycerate, it dephosphorylate and converts the ADP into ATP. That's why it is called, as this phosphate group is utilized or is taken up from the substrate, that's why it is called a substrate level phosphorylation. So by dephosphorylating the 1,3-bisphosphoglycerate makes the 3-phosphoglycerate. Then the position of the phosphate group is changed from carbon number 3 to 2 to make 2-phosphoglycerate, and this reaction is mediated by phosphoglycerate mutase. Then the 2-phosphoglycerate is changed to phosphoenol pyruvate. So here actually a double bond is created between this carbonyl carbon and this carbon, and this reaction is mediated by enolase. And then at the end the phosphoenol pyruvate is converted to pyruvate. So what happens basically, it dephosphorylates. So when it dephosphorylates, again the phosphate level, substrate level phosphorylation it occurs, and the phosphate is released by the phosphoenol pyruvate is taken up by the ADP to make the ATP. So this one is a complete overview of the glycolysis, the glucose molecule. The payoff, this one is the preparatory phase. The glucose molecule, with the help of the hexokinase, it is phosphorylated. The last carbon is phosphorylated to make glucose 6-phosphate. Then this glucose 6 phosphate is converted to fructose 6 phosphate. What happens? This aldehyde functional group is changed to ketone functional group, and the reaction is mediated by a phosphoglucose isomerase. Then the fructose 6 phosphate is changed to fructose 1,6 biphosphate by the phosphorylation of carbon number 1 and carbon number 6. Then fructose 1,6 bisphosphate is split down to make two 3 carbon compound. One of the 3 carbon compound is a hydroxyacetone phosphate and the other one is glycerol phosphate. But undiluted dihydroxyacetone phosphate is converted to glycerol 3 phosphate. The glycerol 3 phosphate, from here this point onward the payoff phase starts, because here they make the energy rich compound. Glycerol 3 phosphate is converted to 1,3 bisphosphoglycerate by the phosphorylation, and here it also reduces the NAD to NADH. Then the substrate level phosphorylation happens. So the substrate which releases the phosphate, dephosphorylation, upon the dephosphorylation, it is taken up by the ATP to make the ATP, and the 1,3 bisphosphate glycerate is converted to 3-phosphoglycerate. 3-phosphoglycerate is then converted to 2-phosphoglycerate by changing the position of phosphate group from carbon number 3 to carbon number 2. Here you can see that it is shifted from carbon 3 to carbon number 2. And then a double bond is created between this carbonyl carbon and this carbon to convert the 2-phosphoglycerol to phosphoenol pyruvate, and then again the substrate level phosphorylation occurs, which converts the phosphoenol pyruvate to pyruvate, and this one is the last step. So this is a general overview which decides the fate of the glucose molecule. So glucose molecule, it undergoes initially a 10 successive reactions of the glycolysis to make the two molecules of the pyruvic acid. Then it depends, in the presence of the anaerobic condition, the pyruvate molecule is converted to either a fermentation process to make the ethanol, or in the anaerobic condition it undergoes a different stepwise metabolic reactions to make the lactate, or in the aerobic condition it makes the acetyl CoA, and then the acetyl CoA joins the Krebs cycle to make carbon dioxide and water. Those diseases or disorders which are associated with the dysfunction of the glycolytic pathway, and all these disorders are caused due to the dysfunction of the enzymes which are mediating each and every step of the metabolic processes to convert the different intermediates into the other one. So here these are the list of the different enzymes and their associated conditions. For example, the hexokinase, it leads to a rare autosomal recessive condition, and the symptoms which are associated with the dysfunction of these enzymes are usually non-spherocytic hemolytic anemia. Phosphoglucose isomerase, it also causes a rare autosomal recessive condition, and the symptoms involve hemolytic anemia, and very less symptoms of this disease involve the neurologic conditions. Phosphofructokinase, it leads to a glycogen storage disease type 7. It is also a rare autosomal condition, and it leads to myoglobinuria, hyperuricemia, hemolytic anemia, and the different conditions. So this condition may be managed by avoiding the high carbohydrate diets. Aldolase, its dysfunction leads to rare autosomal recessive condition. There are three genes, ALDOA, which express mainly in the muscle, so that's why it affects the muscle. The other one is ALDOB, which express in the liver, so it's affect the liver and some other tissues like kidney and the intestine. And ALDOC, which mainly express in the brain, so obviously it leads to neurological conditions. Triphosphate isomerase, it causes the rare autosomal recessive condition, for example, congenital hemolytic anemia, progressive neuromuscular dysfunction, susceptibility to bacterial infection, and the cardiomyopathy. Glycerol 3-phosphate dehydrogenase, it causes the rare autosomal recessive condition, and very little information is available with respect to the dysfunction of this enzyme. Bisphosphoglycerate mutase or phosphatase, it causes a rare condition, for example, hemolytic anemia, polycythemia, increased hemoglobin affinity for the oxygen. Phosphoglycerate kinase, it causes rare X-linked condition, means the gene for this enzyme is present on the X chromosome, and it usually causes the chronic hemolytic anemia, myopathy, with muscle symptoms, especially upon the exercise. Phosphoglycerate mutase, it causes rare autosomal recessive condition which mainly affect the skeletal muscles. Enolase, it also causes a rare autosomal condition which affect the muscle and causes the exercise intolerance, means the person is unable to make the exertion. Pyruvate kinase, it also causes autosomal recessive condition. It is mostly inherited, and the symptom associated with the dysfunction of this enzyme involves hemolytic anemia, jaundice, fatigue, dyspnea, tachycardia, and splenomegaly. Lactate dehydrogenase, it is also a rare autosomal condition, and mainly there are two conditions, LDHA and LDHB. So the LDHA mainly involves the skeletal muscle, while the LDHB it mainly involves the heart muscles.